able to stay through the event, but please feel free to, if you're not feeling very well, please feel free to let us know, and we can save the questions for later.
Thank you. Thank you for understanding.
Of course. So now I just want to address the audience here. This is a very open opportunity. It's a very rare opportunity you have here. So please, I urge you to make the most of it and add in your questions in the Q&A box or get it answered live from the panel of experts here. So please don't waste this. So welcome and let's dive in. So I want to start off by mentioning how much the current R&D in the world around cultivated meat is really raging. We have all witnessed the GFI's State of the Science global webinar, and the companies and academic laboratories, they're all just pushing the boundaries of cultivated meat research. Keeping this in mind, I want to invite Dr. Rathnesh to talk more about what you believe is really vital for the upcoming opportunities in cultivated meat. So please, I invite you to share your screen and present.
Thank you, Chandana. And I have a little, 3 or 4 major important points to make on this particular area. So allow me to present this, and then we can talk more on those topics or the topics which has been a prime importance for today's program. Please confirm if you could see my screen.
Yes, we're seeing it now. Yes.
Okay, so thank you everyone for coming for today's program. As Chandana rightly mentioned, that I largely came from the area of biopharmaceutical industry. Considering we have a food department here, so food is something which is we are seeing, but neither we are food scientists or not we have the expertise in food science area, to be honest. However, considering the cultivated meat industry is growing pretty fast, and some of the areas which connects to culture are also connecting certain dots, and the learning which we have had in this particular industry, we would like to share. So cultivated meat, my perspective today is coming from biopharma, which the startup who are attending today might be important for all of you. Before I begin, just would like to tell you about ICT, one of the unique institute in India, set up in 1933 to help chemical industry. Our inception, our genesis is to support industry and support entrepreneurship. We are also one of the unique institute who handles entire chemical technology, which is food technology, polymer technology, dyes technology, fermentation technology for years together. We also run some of the best biotech programs in India and one of the biggest feeder to the industry in terms of entrepreneurs as well as employees to the organizations. We're also hosting the first generation chemical entrepreneur of our country, and responsible for most of the industrial development have happened between the western side of Gujarat and Maharashtra. This is a list of a couple of alumni, a lot of motivation for all of us to work in the area. So this is very, very simple list. We have around thousands of such alumni who are change makers from industry side or academic side, or also the other areas, whether it's finance or report journalism, or even also motion pictures. So there are set of alumni who are significant amount of motivation to all of us, and that is also allowing us to do something different. Coming to our research group has 2 divisions: pharmaceutical biotechnology and biological characterization lab. Our labs largely works on the 2D, 3D cell culture, spheroids, 3D bioprinting, and organ on a chip, which is related to pharmaceutical biotechnology lab. Biological characterization lab has cell line development, upstream bioprocessing, analytical characterization, scale up and product development. Now coming to the question which may be essential for today's experience. So couple of prior learning, and these are not our own learning, this is a global learning across the world. So any biopharmaceutical development is actually centralized around a stable cell line, its performance. Cell is a host, cell is a genesis, and cell is most important towards understanding any product you are developing. The second important point is chemically defined media, which we call CDM, or sometimes serum-free chemical defined media, plays an extremely important role in product development. So cellular metabolism, if any of the startup, I assume that are Indian startup, it's like Chakravyuh, and it require in-depth understanding before product development. So though you might have studied biochemistry in your previous experience or previous studies, the glycolysis or Krebs cycle, it remains one of the bottleneck towards understanding media and its role, specifically in the cellular metabolic cycle. Going ahead, target product profile and target process parameter. This is very important. The terminology may be very simplistic, but the demand it carries is huge. Unless a definition of product is there with us, or a process and product both are there with us, one cannot define the early development phase. Also, the large scale operation should be visualized as early as possible, because any mistake or any misstep could be very risky and very expensive, considering we are dealing with cell culture. So with this particular experience, when I was interacting with GFI, cultivated meat, a couple of my students started working in this area, and we have realized that how can we develop. So around 7-8 years ago, GFI along with our food department came together and they were trying to set up something together, and that has got our attention that okay, this is something related to cell culture, whether we have any relevance. Because this is something which is very interesting to us. Anything which begins with cells is very interesting. But there are some caveats and there are some other experience as well which is needed there. So cultivated meat, which comes from that particular experience of biopharma which we are having. The same question goes here as well. And if you see the current achievement of good meat, which is the only FDA approved meat product, if you could look into that particular achievement, that also gives a good guideline as well as mandate for most of the startup to follow that path. And also there are a lot of challenges which are going to be there. So one of the most important challenges which we are seeing that remains there in the other industry, that will also be remaining there in this industry, the cell line development and the cell which is going to be derived from animals. Now the characterization is development. If you look into Indian perspective, most of these cells are coming from cell culture collection, which is either American type cell culture collection or European cell culture collection, or there are other UK or Korean cell culture or Japanese cell culture collection. There are multiple cell culture collections. The cell requires significant amount of development. One could also do their own cell culture development. However, it's a time intensive and resource intensive requirement. But this process, you don't have to re-engineer. It is very, very well established. How the cell line has to be established, how it needs to be characterized is already established. Where the innovation is needed is the focus on animal cell. If you're looking for bovine cells or chicken cell or any other kind of animal cell, that is something which is key requirement, and getting the right cell is maybe just 20% of the job, but characterizing it, making sure that it's free from viral infections or any other possible impurity generation, that requires significant amount of time and resource investment, and also validating the same. Media and upstream process development, again, one of the most challenging step, and also a successful one would actually give assurance to the right cultivated meat development path. So upstream development is a first proof of concept that your path is right. It means your cells are growing, they are developing, they are diversifying, they are, creating the right kind of tissue. Media, again, has been considerable amount of development worldwide. Still, media as a requirement, but media per se has been developed significantly in terms of uh, if you look into 6 or 7 major companies which have developed media, this particular set of development have good guideline for all of us. Defining cultivated meat products, again, the TPPs would be essential because cultivated meat is in the first 2 or 3 steps have some relevance to the biopharma industry. But the later part where you are going towards product development, it leaves the biopharma domain. It enters into the traditional food and fermentation technology domain. It means you might be looking in 3D bioprinting, you would be looking into extrusions, or you will be looking into product development, making it that right kind of nutrients, the requirement of essential vitamins and proteins are there. It means it will actually factor into the food development process. So a startup has to look into multiple aspects. One is a biology, which is the defining step for products. On the other side is a product development, which is a very different industry. Just to tell you, if you look into the food science and the food scientists coming from country, there are very limited food scientist labs. So understanding food as a food product would be a very, very different ball game altogether. So it's a cocktail of expertise, which is very difficult to match. So you need to be very focused on innovations as well as focused on collaboration. There are a lot of support you will need at each step. Again, depending on what is your product. If your product is food, then definitely, that's the end game. But if your product is cell or a media, then you are only looking at that particular side of the story. Again, a startup should also focus on supply chain challenges like cell line development. It's not easy. It's one of the difficult and also one of the most rewarding one. If you actually can develop your own cell line and your own systems, it's extremely revenue rewarding, and in terms of the amount of money one startup can generate for the right kind of cell line, and the kind of impact you can bring by providing right kind of food component, this would be huge. So this is one of the gray area and also one of the gap area where startups can focus. Media and its components. So media alone have 450 components. So it's a very complex mixture of chemically defined components. But it's component and also some of the right kind of a growth factor, right kind of so each animal cell will behave different. It means there is no default media for a default cell. If you visualize that I have a media I have developed and it is going to work for all the cell, that is not true. You need different kind of media and its component. So the basic media will remain same, but the feed which is going to be given to the cell, it would be different. It means it also gives again an opportunity in the area for many startups to define what kind of components they might be working. Extruder in food industry is a very common thing. But when it is going to look into the biological, the cultivated meat industry, and the kind of food it is going to come, the kind of muscles or different kind of tissues the cell is going to bring, you will require a very different kind of extrusion requirement, which is very, very limitedly explored. Not too many people, hardly there are 3 or 4 worldwide groups which are working in this area. Bioreactors, of course, this is again a very much engineered, the growth in bioreactor is huge. So there would be only certain modifications or certain requirements based on the cell would be needed, but largely scaled up bioreactor along with aseptic requirement would be a key area. Sensors and process solution and many other characterization tools to make sure that cells are growing, cells are making right tissues, and also any kind of toxicity or safety issues are not there. These are the tools which are gap areas in innovation, in my opinion, can be a right perspective for most of you. So last take home message, all the affordable drugs and vaccines are developed in India. So whenever you look into the most competitor economic solutions, India has given the answer. It has demonstrated that and continuously growing towards affordable drug development. I'll just give the same replica to cultivated meat industry, which is a very niche area as of today, but it will need significant time, knowledge, and resource investment. But at the same time, Indian innovators or India as a country would be essential towards affordable development of cultivated meat, which can be very expensive if wherever it is being developed at the moment. So that's a message, and if we innovate right, pain points, we'll have a good amount of development in future as well. Thank you very much.
Thank you so much, Dr. Jain. That was definitely you covered everything that we were envisioning should be talked about in this particular white space area for cultivated meat. So thank you so much. And just to follow up with that, I love the analogy you gave between the vaccine manufacturing industry and how similar cultivated meat and fermentation sectors can be, because in the end, the biomanufacturing tech are very similar, if they're not very different at least. So what would you think? How has the research and entrepreneurial ecosystem in India, including what the subject matter experts in India think, and what do the policymakers think about this field's potential? Like, I know there are so many entrepreneurs here right now wondering the same thing. Like, fine, we recognize that this is a very up and coming area, which is very vital for securing the food future. But what do you think the general interest is among the high level policymakers, for instance?
So thank you, Chandana, for this question. I think the kind of development, as you might be aware, and this is one of the priority area which is being considered at the policy side. Within our circular bioeconomy initiative, there would be a good amount of focus would be given largely to biomanufacturing. So when I'm saying biomanufacturing, food is just one of the part. There are definitely other biomanufacturing initiative which will be coming up. So positively, from as far as policy is concerned, there is going to be a good amount of policy. As far as regulatory development is concerned, which also relevant to the policy, I think FSSAI have taken positive step, but I think once the science is developed, regulatory development will go hand in hand. And considering Singapore or US or many other countries have positive regulatory development, which will also allow policymaker to create that industry. But at the same time, what is key and required even for a policy or a regulator to come up is demand. Unless there are good amount of demand or innovator startups are created which they are demanding, the policy won't come, neither regulation will come. So I think it's a simple demand and supply rule. We should not expect that policy will come and then startup will grow. I would advise reverse, that let the startup grow, policy will come automatically.
Completely agree. Yes. And Madhuji, Dr. Goswami, if you have anything to add here, please, you're welcome to.
Yeah, I think quite a few things have been kind of stated about the status. But what for the startups, see, if you look at the clean meat context, it's a very long process. And there are a number of them, maybe several tens of challenges that one can look at it. And I am sure that no single startup can really looking for end-to-end kind of a thing. Here, only that's one thing interesting about it. There are so many opportunities that are there for startups to solve. And these solutions is what everybody is looking for, whether it is, how to increase the number of divisions, or how to make sure, of course, media challenges are everywhere. How to come up with the media, because media is supposed to be one of the most expensive component in the production. So can we make it more affordable? There is so much of new things can come up, and the innovation can be made. And the other thing is, whether it is scaffold, or is it the surface modifications of the scaffolds, or or even in the fermentation process itself. I mean, I can go on. There are enough number of challenges where startups can contribute easily. The only thing is they have to identify, say, for example, in recycling of the media, for example. So because that's the most expensive one, can we recycle it in a way so that we can cut down the cost in a big way? Because in the end, definitely any technology is looked at. What is the cost benefit analysis, not only in the not only in the financial terms, but also environmental terms. So if you look at in those contexts, these kind of things will help a lot in making this particular technology take off. And the general justification of going towards this clean meat kind of approaches is kind of a given thing. It's kind of written on the wall. Everybody knows what needs to be done. It is probably, we definitely have to pursue it, because we can't really start doing something when you really get into a danger. So it's very important. And there are so much of work already been done. And also GFI also has been extremely active in identifying the problems. In fact, I really enjoy reading their website, where the challenges are very, very well stated. And that also gives a lot of opportunity to startups. But if you look at one of the best things I heard in last 15 days about in this context was, I was attending one of the DBT meeting where the DG was also speaking about one of the 6 things that they identified in bioeconomy was smart proteins and clean meat. That for me was a kind of a music to the ear, more because uh, since they invested some money in our project at CCMB, there was no further government activity in that. Like, obviously, a clean meat is not something that can be done in 1 or 2 years. So this is first very good emphasis that DBT has come up with. That means that DBT is going to look up at creating a kind of center of excellence, or a kind of separate vertical for funding these things. So that's a very important news that is coming from the government. As far as universities are concerned, or academia is concerned, I don't think nobody is really looking at it in a very in a clean meat context. Probably they have their own solutions that can contribute to clean meat. But these people come out of the wood once there is a kind of a grant money, a grant vertical is established by the government. So I'm sure once that happens, a lot of people come and participate, and the whole thing will take its, it gets accelerated to the next level. As far as startups is concerned, there are not many to write about, but I really give a big hug to all these guys, because they took a step in that direction, and I'm sure their judgment is not wrong. But my request is they should look at a problem at a time, because they cannot really solve end-to-end, whether it is scaffold, or it is recycling of the media, or something else, or how to make the cell lines non-GM kind of a thing, take best precautions to make it non-GM kind of a thing. These are the kind of challenges that are actually accessible in terms of technology. Most of the entrepreneurs or the PhDs, whatever that we have around in this country. So in my opinion, it's a great opportunity, and one has to believe on all these, the promise that these kind of technologies have down the line. I'm sure there will be an inevitable situation down the line to adopt these technologies. The West has been they are doing it for various reasons, but we have to do it for our own reasons, which is something that not only environmental, because that's the big threat around, but the other part is that we can do, because it's not something very difficult for us. It's not rocket science that we can't afford. All of us can do, in the sense that in every lab in the country probably can contribute something to this, but they have to believe in the larger goal. That's what I think. I'm sure, I mean, more people come and ask, we can address those questions. Thank you very much.
Thank you, Madhuji. Yeah, I think you nailed it there by saying it's as interdisciplinary as it is. We have all the tools at our disposal, whether it's biochemistry. Now there's AI, computational biology advances, which we can use to do spent media analysis, flux analysis. So we do have it. Every lab, every scientist in India probably has the ability to do it. Resources are lacking though at the moment. So that is something that the government is taking the step in the right direction. So we're hoping support will only grow for our scientists to also grow in the sector. So that perfectly summed up all the white space opportunities that are out there, including cell line development, media analysis, media, because like you said, cost is majorly driven by the media. So if that shell is cracked, then I think we're halfway there. So that said, I think we can, as an active researcher, I want to talk to Dr. Goswami about how he is actually using non-GM approaches, in fact, to establish fish cell lines from native species of fish. So Dr. Goswami, what do you think? We know that a lack of readily available cell lines is a huge bottleneck, and startups aren't trying to do end-to-end, like Madhuji said. We have to focus on particular white space opportunities in the pipeline. So what do you think is still unexplored in India, and what are the bottlenecks to help increase more research and advancement in cell line area?
Yeah, in my opinion, cell line development, as from my experience, if you see as a sector, this is an alternative food sector. So if you are going to grow a food sector, so all kind of integration will be required, starting from the raw material and reaching the raw material, and then end product, and then after regulation reaching to the consumer. So all this together, so it's not a one-man army. You can't say that only the development of cell line is a bottleneck. I think the development of cell line won't be a bottleneck from my experience, because we are doing or some specific cell line, so we are able to grow. Only there are different bottlenecks are there, where the startups can come into, suppose the serum replacement, some algal proteins are there. See where we can look for those, where the startups can come forward. And then again, to grow them in the three-dimensional phase, scaffolds, again, the scaffolds are not readily available on that. So again, some startups can look into that. Again, that scaling up, that bioreactors and all, although we are developing some, but it is not a big way going. And then 3D printing, and then going to the end product. So this, I think, I would like to recall one statement which was published in Nature. The academic research can speed up the process of cultivated meat to the market. The way the GFI is coming up in India, so necessity is convinced, fine. The necessity leads to the invention. That necessity is there. We need to have alternative protein. The 10 billion people will be there. All those resources, whatever fish we are getting from aquaculture or maybe open water fisheries, challenges are there. So we need another alternative. That is convinced. But academic research, government means funding, altogether should come up more optimized right material for product development, and those startups should come forward. So altogether, it's an integration. That to grow a cultivated food sector, all those should come forward. The only I don't think the cell line availability or certain things are not bottleneck. Cell line, yeah. That obviously I think after maybe 1 or 2 years from my lab only I have developed many cell lines. But only problem is their proliferation to get more cells. All those things need to think with serum replacement, as I say.
Yeah,
I think altogether,
getting the right kind of food safe media and eventually helping scale it up in the right way to develop an end product. So sourcing the cells themselves isn't a bottleneck. It's having a well-characterized cell line which is able to scale.
Dr. Chandana, just I would like to add one more point. I am also part of the scientific panel in FSSAI. So FSSAI is also looking for some kind of product. So once some product will come, then only its evaluation will be there. Then only slowly it come to it. So altogether, it should come, then the regulatory process also it will pass. So I don't think so there will be many hurdles. If all those startups, academic researchers are coming together, I think we'll have a very rosy figure very soon. Thank you.
Thank you. That's a very hopeful way to end the first part of this. So just to summarize, cell lines, media development, exploring alternative sources of media, like rather than just going for microbial fermentation derived recombinant proteins, which can become expensive. There are various other ways we can investigate. Like, for example, there is a professor, William Chen in NTU. He investigates agricultural alternate side streams to make serum replacement. Like Dr. Goswami was saying, we really need serum alternatives to make cultivated meat work at scale, to meet the price at scale. So to achieve cost reduction, we really need to look at, like you said, algal serum, like protein derived from algal, or macroalgal, like seaweed derived proteins. These are all excellent sources for us to investigate. And in India, there is no dearth in supply for agri-food side streams, for seaweed species or microalgal species. So these are all opportunity areas for startups to investigate and eventually come out with something that is scalable and would help reduce the price of media. So with that, I think I would like to move on to, we've already talked about food safety and how food safety and quality will, the government is ready to look into this. The recent announcements that they've made have been very promising. So as our entrepreneurs and the industry grows in India, as cultivated meat startups go and apply with an end product, I'm pretty sure regulations would follow. I think Dr. Rathnesh made the same point earlier. So all that is brilliant. I think I would like to now sort of shift gears and talk about something called as hybrid products. This has been something very sought after as a kind of a shortcut to make a better product at lower cost. And to give an example, instead of growing cultivated meat, cultivating cells just on empty, on a scaffold-less or microcarrier-less model, there are opportunities to grow these cells and put them on plant-based scaffolds, which would have fiber and have a good construct for the cells to grow on. So any thoughts from, and I invite anybody here, Dr. Rathnesh, because you are a biomaterials expert. So I'm curious to know what sort of opportunities in India are out there and what we haven't yet explored. Because as far as I know, there is one mycelial scaffold company so far, but otherwise not much has been explored in the area. Do you believe that's something we should be looking into?
Yeah, I think one of the major unexplored area would be natural fiber or natural polymer as well. Though for decades, I probably would say for 5 or 6 decades, such a development is happening. One of the major challenges is availability of purified material. So what cultivated meat demands is a purity level. So which in our industry, we typically carry a GMP standard or something which will have 99% purity level or little higher percent of the purity level. So say I'm just giving example, guar gum, one of the major, major material available with us. But when you look into the availability of GMP grade guar gum or the guar gum which will have lower amount of impurities, it is very difficult to get. Just to give you an example, the challenge, very simple challenge. You all are aware about salt, sodium chloride. One of the key requirement in media, doesn't matter which media you prepare, you need sodium chloride. The surprising fact is that we still don't have a technology to get sodium chloride 99.99% pure. We still have, we are the largest salt producer, but the purity, when it comes to purity, we don't have that tag. So, the technology demand and requirements are enormous and they are just next door. You really don't have to look around. So coming back to your question of biomaterial, there is enough, and we will need. So if you look into 3D bioprinted meat, which also as a product development phase, you will require bioinks. If you need bioink, you need materials. You need materials which can probably replace or supplement ECM components, allow cells to grow, allow them to convert into a more complex set of tissues. So when we are looking into it, there are enormous amount of natural material, cheaper material. What we need is a purity level. If we can actually demonstrate that we can have purity, then definitely that is a way forward. And I think that we should definitely explore.
Got it. And also, is it available at scale? Would it be manageable price-wise? Because as much, so for example, there is something that often comes up in our life cycle analysis of cultivated meat. The ingredients, like the recombinant proteins that go into making cultivated meat, don't necessarily have to be pharma grade. They just have to be food grade. They don't have to be ultra pure for it to be toxin free for consumption. So that is also another area that we should often think about when you're making cultivated meat. It doesn't have to be tissue engineering grade, which is going to go into your body as a transplant. This will have to, because it's very important from a price perspective as well, to make sure that the materials you use are edible, it's safe for consumption, but at the same time, not going overboard with the GMP requirements as well. So do you think that that's a fair argument?
Yes and no. The yes is definitely food grade material. I mean, that's my perspective and the way I'm looking things. The food grade perspective, food grade material is okay if they are being directly consumed as food. But things are a little different here because you are going to use them in culturing cell and making tissues while creating cultures and while creating tissues. Though we are going to consume as eating, I mean, it's not an injection, it's not a drug, but certain impurities may create any allergic reaction or hypersensitivity reaction.
Correct.
And that impurity characterization, though the acceptable criteria by regulatory agencies, they may lower down, say 99 to 95%. But whatever that 4% is, so like just to tell you at current Indian regulatory agencies specifically, I'm not sure how much people bother about each and every 4% or 2%. But if the cultivated meat industry has to grow, I think each of those percentage has to be defined going forward. So that is probably my concern and perspective, that if probably we are characterizing it, we should characterize in one go.
Correct. Yeah. And then later on, according to how the regulations turn out to be, we should be able to adjust for price parity and of course adjust for availability at scale if we are looking to eventually produce this in tons. And now this brings us to like the next point of high contention, challenges related to scaling and commercialization of cultivated meat technologies. And this is acknowledged across the world, regardless of where we are. And like even if we were to produce something perfect at lab scale, how would it scale up? How would the price go with scale? Do we have the production facilities for pilot scale production? Forget about commercial scale, just from lab to demo pilot. Like, do we have such facilities in? Because I'm pretty sure this is on the minds of startups who are close to finishing up their R&D in lab, and then they're like, what next? What do I do next? So Dr. Rathnesh, I know you are working really hard in this area in synthetic biology and biomanufacturing sector. So I would love to hear your thoughts on what are the gaps and how do we bridge that for commercialization and scale up.
Thanks, Chandana. So yeah, I think this is the pain point. In my language, I'm calling it valley of death, and I think in everybody's language we are using the same thing. So any early innovation is going to simply die down if there is no commercial proof exist, and that require the first proof of validation. So in terms of infrastructure, just to tell you, Chandana, our organizations, institutions, Dr. Rao is here, and there are many other institutions. The infrastructure is around. When I say around, it's around. It's not synchronized. That is what I have felt. I mean, in my whole journey of visiting multiple institutes across the country, and almost most of the institute I have visited, major institutes, infrastructure has been established. It has to link, it has to be dedicated to certain cause and purposes. So if I'm looking for a microbial cell culture, I really cannot use for product A, for product B, or product C, and that basically creates a lot of problem. Similar issue exists for for mammalian cell culture or animal cell culture. So when we are looking for, these are much more sensitive, biosafety levels. It means you have to have a dedicated facilities and synchronization. So somehow over the last few years, most of investment have happened in the analytics, in the characterization space, but not in the manufacturing space. So when it comes to reactor capability, there are certain institutions which have certain ability if they would like to remodel their facilities into this space. Again, demand and supply, this probably could create that. At the same time, as I say, that we are also trying to set up an example here of synchronized facilities, that how scale up is a requirement. So at 100 liter, 200 liter, and you rightly said, we really don't have ability. So yesterday I was reading about ABC corporations commercial 250,000 liter capacity for good meat. It's a pretty large facility which they are building in US. But similar facilities can be built as well, but that's a final destination for having such a production capacity. But if there is our pilot scale validation and successes, that is not something which is not possible. That is definitely possible. But again, that 200 liters, so there is enough amount of need. Even the startup can come into the space. They can create not only the innovation model, but service model. Service model is also CDMO growth model itself is going to 14 to 28%. Our in-house company, Syngene, has demonstrated that, such a service model exists and very profitable. Worldwide, there are many service industries who are also doing. Samsung is one of the most successful service model. So service model exists. However, you have to demonstrate expertise. So while just answering your question, we really don't have a dedicated pilot scale setup, but this is being in motion. This is not something, so by the time I believe that early phase of these startup ends, government of India will definitely be going to be supporting certain tinkering or certain pilot scale lab, which can be utilized by these startups.
Yeah, that's true. I think we are getting there as well, because the first step is them identifying this, and of course, given our already present biomanufacturing sector, the tinkering, like you said, the talent, it's all there. It's just about us guiding the right policymakers to make that happen practically. And Dr. Madhusudan, I'm curious, as someone who runs a bioincubator, and I think you have in-house a fermentation, an all-protein fermentation company and a cultivated meat company. So I'm curious to know how they are managing for this pilot scale gap, and what do you think is needed for the sector to grow?
Uh, as- See, most of the initial work, say, let's talk about the clean meat company. The name of the company is Neat Meat. Their initially their interest is to develop chicken cell lines towards meat purposes, and they have been isolating these cell lines and then characterizing them, because one needs to be very careful in characterizing these things. So they are still at a stage where they're in the flask stage, and what we have in CCMB is a 15-liter capacity. So that is also a good deal for a scale-up level, but that's not what Dr. Rathnesh is talking about. But one doesn't go from a flask to a big one, but they have to demonstrate the the cell density, those kind of parameters, which are most important parameters for this thing. And also to, here now they are using not a complete serum replacement. They use some amount of serum, but like 2%, 3% kind of a thing, and slowly wean off the serum kind of a thing. Those kind of experiments, and to demonstrate them at to show it in a fermenter level, I think 50-liter is fine to begin with. But when you have too many combinations to explore in media optimization, this may not be a bad idea to do to begin with. Of course, they'll do earlier in a flask level, demonstrate the appropriateness of that particular composition. So, but the other company which looks at fermentation of milk proteins, they do most of their biology work here, and they also have another setup in Bangalore where they do the scale up. They have up to 50 to 100 liters, but here we are looking at mostly at yeast fermentation, which is something not too many unknowns are not there, like what you have probably in the in clean meat context, because in clean meat you have more unknowns, and also a lot of exploration need to be done to cut down the cost, like what you're talking about, how appropriate to use the food grade components versus laboratory grade materials. So in my opinion, the challenges for fermentation of, say, for example, yeast organisms is kind of a much more solvable problem than the clean meat kind of a thing, where many things are not known. And if you really want to come up with because everybody looks at media, because that's one thing that may make or break the technology, life cycle analysis and so on and so forth. So in that context, the some in my opinion, no single media will suit many purposes. I think you have to go and develop as a pair the cell line, and also along with the media, which is obviously going to be serum free. Whatever whether you add algal proteins or plant-based proteins as a amino acid source, how do you optimize? All these things can be done, but may have to be done in the end as a package rather than a cell line alone versus the media alone. In my opinion, you are not going to ever get a media that is useful for different cell types. I mean, different cell origin cell types. I mean, like, chicken versus fish versus this. So there has to be a lot of customization, which probably is good because the company can claim the proprietary, and then there is a proper intellectual property to it. So media engineering and cell development should probably go hand in hand, or so people can collaborate. So somebody has a good cell line, they should share their cell line to those people who are developing the media and has more feel for what can be done. So in my opinion, I think that's how it should be going.
Got it. Yeah, yeah. So we are still in the early R&D stages, and that said, even if there are people looking for scaling it into demo. There are CMOs based in South India which will gladly help them out, whether it's animal cell culture or microbial fermentation related scale up. So for now, the facilities are sparsely distributed across India, but it is there, and I would encourage everyone to reach out to GFI India for such contacts. And an interesting analysis came up recently about manufacturing capacity and how retrofit can potentially solve problems about, rather than building new capacity, can we retrofit decommissioned bioethanol facilities or biopharma facilities which were doing vaccine manufacturing and have stopped, or breweries, etc. So this is another area of exploration, rather than spending a lot of capital on building new systems. The government will be looking into retrofitting for making it suitable for cultivated meat or fermentation related biomanufacturing. So yeah, for pilot scale and demo, I think we are on our way there to be more accepting of commercialization pipelines. So with that, I think we have managed to cover a lot, right from cell line, media, scaffolds, all the way to scale up challenges. Maybe we can start moving on to some questions that have come from the audience. Let's see. Dr. Satish Kumar Singh is asking, thanks for the presentation, Dr. Jain. Do you have recommendations for obtaining cell lines for mutton and chicken in India, either from CSIR or private organizations? Dr. Rathnesh and Dr. Madhusudan, I think both of you are well positioned to answer this. So please go ahead.
Yeah, so in my opinion, the sourcing at the moment should be done from recognized cell culture collection, considering the data. So whenever any cell goes for commercialization, one need a documentation as well as licenses. It would be advisable that one can get even a research license, and eventually when there is a commercial success, you could convert that to a commercial license. I would discourage any startup to get cell licenses from any laboratory. There have been incidences and evidence where the company has grown significantly at commercial level, and when there was a cell line being investigated, the cell line was found to be donated by a lab and ultimately deter the chance of regulatory approval. So in certain cases, please avoid getting cells from any partner lab, helping lab. It has to be a commercial research license purchased by you, and then only. So these are cell culture collection. Just to give you an answer, find out the right cell culture collection and get things done from official licensing process.
So I will add something more to it. Is say, for example, what we have been doing at CCMB is isolating taking the biopsy of the animals. In our case, it is sheep, and then isolating the stem cells, because all the cultivated meat depends upon stem cells. Obviously, the beginning is stem cells. So one has to characterize these stem cells about their ability to become differentiate into myoblasts, myofibers, and things like that. So, and also characterize them very carefully for all the molecular markers like whatever myogene, myod's, and then px and all that. So somebody who does all that and then gives, say, how many generations it will go on, some kind of a genomic stability analysis, and then I think that is something is where you would go, because some of these cell lines are not commercially available and they have to be characterized individually in the labs. And then that is something is fine to take, and if somebody is interested in the next step, that is, say, if somebody is interested in media optimization, they should go for these kind of whether it is academic institute or the commercial, it doesn't matter. So but basically somebody who had done the characterization very well. Probably these are not yet approved by any of the culture collection things. And in fact, it is the product that somebody can sell. also, in the sense, if I have half a dozen cell lines with me, I can get into a kind of a license agreement with a media developer or a fermentation specialist. So it's fair. I mean, like, what comes to you, you should be able to reproduce what the giver is claiming. Like, if somebody says this goes to these many number of divisions, I don't have any problem with the thing, and I didn't do any genetic modifications, and the genomics is clean and all that. If somebody gives me, and then I reproduce all the markers, then I think it's fine. I can go ahead and do it, because these are not going to be commercial. Nobody's going to commercially available, but not not from any of the culture collections, in my opinion, not necessarily.
Got it, got it. And if we are eventually looking at a commercial application for those cell lines, one thing we've learned from all the FDA documents is that they want a solid paper trail, right from where did you get the biopsy of the from,
Yes.
what was the animal's health condition. So everything has to be very micro documented. So if you're just doing something for cell line, media optimization, then you probably don't need such a solid paper trail. But otherwise, if you are looking for commercialization eventually, it's very important to remember to get it from certified labs, get it certified for any microbial contamination free, viral free, disease free. So all these need to be from government certified, lab approved. So this is very key for eventual commercialization. But if you're doing media optimization, etc., like Madhuji said, it's important for you to know how well characterized the cell line is from a reliable academic or an independent lab. So you know for sure you know what you're getting. And I think Dr. Goswami has left, but he does this with the cell lines. He has developed in-house fish cell lines. He does partner. You sign an agreement, partner with other startups, entrepreneurs, companies to develop such media using his well-characterized cell lines, which again he has the entire documentation of where the cell line came from, the origins. So there are such matches you can make. Satish Kumar, if you need such matchmaking, please contact us and we'll be happy to help you find sources of cell lines. Thank you for your question, though. That was very relevant. So moving on, Satnam Singh is asking which option appears to be more scalable and feasible for cultivated meat production. Is it adherent cell culture or suspension cell culture? I would allow either of you to try and address this.
What I mean, I don't have ready answer, but what I can say based on the experiences, it's more to do with the specific cell type, what is its adaptability to be a suspension cell versus and also adherent thing, and how good is the scaffold. In the end, it is going to be, again, we keep coming back to one particular arbitrator, that is the cost. So to what extent, what kind of densities one can reach, and sometimes, like what Dr. Rathnesh was mentioning about, is the scaffold can be part of your final product. If the scaffold is going to be final product, so it becomes, say, many of the cluster bean proteins or soybean proteins and things like that, or even algal proteins, they can be scaffolds for you. So it's a kind of a scaffold plus your protein, that's a cell line. These two can contribute in the final product. So that's another way of looking at it. So in my opinion, today, a clear answer for you is not going to be there, more because it is a combination of these things, your own company goals, all these things do come into play. So definitely suspension cells will go more density theoretically, I mean, obviously, compared to the scaffold ones. But if your choice is going to be a product where your scaffold is also eatable, then naturally you will compromise on the amount of protein that is there on the scaffold. So these are all something very product specific technology that you need to come to. So that's what I think. So while generally I can say that suspension cells will definitely grow to more density, and you'll get more protein out of per ml or per liter. But as I said, it is the product that defines your choice.
And Dr. Rathnesh, I welcome you to add any comments.
Yeah. So I think Dr. Rao has covered most of the things, which is also one of the way forward. It's all about TPPs, your target product profile. When it comes to suspension cell, just would like to add that large amount of biomanufacturing fundamentals as well as infrastructure is developed for suspension culture. It means while scaling up, you will find limited challenges, considering the bioreactor and any further processing has been considerably developed. While looking at adherent culture, there are challenges, considering this area is still niche amongst niche. So one has to really look into all justification as well as a lot of product development and will go in the process. Again, it's not a deterrent, it's opportunity, but we have to look what kind of product you are developing. As Dr. Rao said that scaffold is something which is key requirement. But of course, if you're looking for a faster progress and seeing what you can work around, how you can develop a product faster, or like to have a learning, suspension culture overshadows the adherent one.
Correct. And looking at what already scaled up companies are doing, Upside Foods or Good Meat, some of them are doing scaffold-free approaches where they have spontaneously immortalized cell lines growing in suspension in bioreactors, and they're able to kind of form a lump of meat which has more of a chicken breast texture, I'm told. So it depends on the type of, are you trying to make whole meat cuts? Are you trying to make just minced meat? So all that will depend on whether your cell line will be adherent or suspension. Like both our experts just said, it depends on your end product. And I would highly recommend you go through the FDA dossiers for both the FDA approved cultivated chicken products. That'll help you get an idea on what's really working for them at scale, and sort of help you understand what the best approach might be. So just an idea out there. Moving on, Dr. Goswami, there's a question for Dr. Goswami, but unfortunately he had to leave. Thank you for your valuable insights, Dr. Goswami. We are very grateful that you participated despite your bad health. Yes, we are. Are the cell lines for Rohu developed by you available commercially? So I can answer on his behalf. They aren't available commercially yet, but if you are interested in entering a sort of an agreement with him to test these cell lines, you are welcome to do so. Please reach out to him and ask him if he's willing to share some of his cell lines for your testing purposes. I'm sure he'll be open for that, for such collaborations. Moving on, what are the regulations we need to follow for selling cultivated meat from edible biological sources in India? So as of now, I think we had covered this in our earlier discussions. As of now, FSSAI doesn't have any regulatory framework for cultivated meat products. So it's a bit of a black box. We don't know. But what I can say is GFI India is working very closely with them to create a scientific panel meant for cultivated meat and other types of smart proteins. In fact, Dr. Goswami is one of the FSSAI scientific panel members, and pretty soon, we hope maybe in the year or the next 2 years, we will have a dedicated cultivated meat panel where we will set the regulations of what's needed to develop cultivated meat products in India. So it's on now. It's on the way. I would say. Please hold tight. As of now, if you are I don't think we are that close to commercialization yet. But if you are looking to, to suit regulations right from the start, refer to the Global Codex Alimentarius on cultivated meat. I would highly recommend. That will give you sort of a rough framework on how to approach for novel inputs for creating cultivated meat. So that's the best I could answer. I'm not a regulatory expert, unfortunately. GFI India does have regulatory experts. So if you want to have a more in-depth conversation about regulations, you're welcome to reach out to us. And last question, cultivated meat can be from sourced other than animal origin. So cultivated meat is meant to be a replacement for various animal derived proteins like meat, even dairy proteins. All of these have to come from some animal source like chicken, goat, sheep, fish, shrimp, etc. So I'm not sure if I'm understanding this question correctly, but yeah, your source has to be from animals. The biopsy would ideally come from a young animal from its muscle tissue to grow the stem cells in the lab in vitro. So yeah, that is how cultivated meat is meant to be. And if I'm sorry if I misunderstood your question, but please write to us eventually if I didn't answer it properly. But thank you for all your participation, and thank you to Madhuji and Dr. Rathnesh for patiently staying through with us for all this time and answering all our questions. I'm really, really grateful on behalf of our entire GFI India and ISPIC team. Thank you so much for, for dedicating your time and energy for this. Thank you.
Thank you for organizing it. And then, yeah, hopefully our engagement will nudge some of these guys to get into a more formal,
Yeah,
get into a more formal activity.
that's
Yep.
the hope. Thank you. Thank you. And Dr. Rathnesh, please, if you have anything to say.
No, thank you, Chandra. I think it's a good initiative. I think all the best for these efforts you all are taking. I think more and more startups should be encouraged and make them aware the challenges. So I think, yeah, thank you very much for organizing this.
Of course. Thank you. Thank you for being here. It wouldn't be possible without your presence. So thank you so much. And
thank you.
yeah, I think we'll wrap it up for today. Thank you and have a good evening, everyone.
Same to you.
Yep.
Bye-bye.
Bye.
Bye-bye.
Bye.
Bye, Dr. Rao.